Alright, hello everyone, my name is Craig Carter and today I'm going to be presenting CRISPR-Cas9 mediated gene editing in human triponuclear zygotes. So CRISPR stands for clustered regularly interspaced short palindromic repeats, and it is a strand of DNA found in genomes of bacteria and other prokaryotes. It consists of repeats of short DNA segments, usually 20 to 40 nucleotides in length that are palindrome. These DNA sequences consider spacers that separate each DNA sequence, with each spacer being unique. CRISPR was actually first identified in E. coli, and it acts as an immune system bacteria as it helps defend the DNA of attacking viruses. Using CRISPR, we can create a system where it can locate a mutation in DNA, remove it, and replace it with the correct sequence. Not only that, but it can also repair needed cells and accurately cut DNA. CRISPR was adopted alongside with the nucleus Cas9, which recognizes DNA sequences. Labs were able to demonstrate that CRISPR-Cas9 could edit human genes in vivo. For the experiment I'll be talking about today, and the techniques used, the labs used the human B globin, which is the HBB gene. So this right here is this diagram shows the techniques used to conduct their experiment as far as CRISPR goes. So using online tools, they developed, designed, and generated 3 guide RNAs that targeted different regions of the HBB gene, labeled them each one G1, G2, and G3. Out of the 3, G1 and G2 gene RNAs produced efficient cleavage after conducting a T7E1 assay. The remaining gRNAs were assigned specific PCR primers for the top 7 predicted off-target sites in the genome. They once again conducted a T7E1 assay for off-target effects, this time in human embryonic kidney cells. When G1 did not exhibit any off-target cleavage activity, it made it the prime candidate for future studies. Next, they synthesized a single-stranded DNA oligonucleotide donor template that encoded for 6 silent mutations. They infected the oligonucleotide both along with the G1 gRNA into human embryonic kidney cells. DNA was then extracted 48 hours later for PCR amplification of the G1 target region. The PCR products were then subcloned into TA cloning vectors for sequencing analysis. After 29 independent clones, 14 clones, which was 48.3%, perfectly matched the donor oleo template, indicating precise editing of the HBB locus. After this, they began investigating the efficiency of the gene targeting in human triponuclear zygotes, which I'll explain later in the next slide. So, triponuclear zygotes contain one egg cell and 2 sperm cells. So for them to conduct this study, they had to they received these embryos from local clinics, because whenever there's a triponuclear zygote, usually these embryos they don't survive, so these embryos aren't born into babies. But the G1 was co-injected with different concentration combinations of Cas9 mRNA, GFP mRNA, and the single-stranded DNA oligonucleotide into the cytoplasm of these zygotes. Based on the morphology, roughly 80% of the embryos remain viable 48 hours after the injection. So, and out of those, all GFP positive embryos were collected. So GFP positive, that means that out of the ones that were injected with and survived. The GFP plus is a fluorescent dye. They were able to detect which ones were successful. So all the ones that's listed on the GFP plus, those numbers indicate all the ones that were an actual successful injection. So after all the GFP positive embryos were collected for whole genome amplification, as well as PCR amplification of the G1 target region, of the 54 remaining PCR amplified embryos, 28 were cleaved by Cas9, producing an efficiency of roughly 52%. To determine the off-target site effects of CRISPR-Cas9 in these embryos, the top 7 off-target sites were examined again, plus the site in the HBD gene. The T7E1 assay only revealed off-target cleavage in G1OT4 and G1OT5. Of the remaining embryos, 4 were paired using the ssDNA ugleo as a template, and 7 were recombined with the HBD gene. Those 7 were amplified and examined. The HBD locus in 5 of the embryos remain intact, containing no HBB sequences. The lack of bidirectional sequence exchange supports the idea that the gene is repaired mainly through non-crossover HDR. But basically, after conducting their research, just by looking at the results, it was easy for them to see that, CRISPR. It was easy to see that CRISPR still has problems with targeting the correct DNA sequences, and correcting it, it would often miss targets. So, in the data from this experiment actually proved that CRISPR still, needs work. It's still missing its target, so it needs to be improved to where it can actually find the correct DNA sequence, correct it by cutting it and adding the new DNA sequence to it. And then once that's fixed, then CRISPR and Cas9 together will be a success. So my question is, according to the data obtained from the experiment, CRISPR still has problems with missing its targets. What techniques can be changed or added to help improve the accuracy of CRISPR, identifying and cutting the correct sequences of DNA?